very happy to introduce Monica Phadnis, who is the VP of Clinical Operations at Metabomed. Monica brings 16 years of clinical oncology strategy and operations experience in biotech, pharma, and CROs, and in academic settings. In her previous roles, she has provided leadership in integrating innovative science with clinical operation strategies in precision medicine treatments, immunotherapy, antibody drug conjugates, and targeted therapy compounds. She holds a BSc in mathematics from the University of Mumbai and a health sciences diploma from Harvard University. Monica is currently working with Metabomed as VP of Clinical Operations.
Thank you, Dr. Tata, and thank you everyone for joining me here today. First of all, technical glitches are where everybody can see my screen.
Yes.
So welcome everyone. This particular topic, Tepotinib and patient enrichment, is very near and dear to my heart. And the main important thing about this is that whatever challenges we faced with this program 4 or 5 years ago. So this program began around 2015 in the preclinical stage. And then throughout up until earlier this year, we were still progressing through this program. The striking point here is whatever we experienced 3 to 4 years ago in terms of challenges and then the solutions are all applicable today in the new norm of post-COVID era. And that's the reason why I selected this particular program. So let's dive into this. So far I'm really enjoying all the presentations and I see a common theme. So all of us here, we know what are the common axes of any clinical trial, right? And the interdependencies where we have the budget, the timelines, and also the competition. But then we have the patient selection and then the trial design and translational medicine program. The reason I'm addressing these key challenges is in the background and in the forefront, we had Tepotinib, which was a lung cancer program, and it was exploring a very, very specific stage 3, stage 4 advanced lung cancer patients, which most of them had c-Met exon 14 skipping alterations. So usually in rare diseases where we know either there is a fusion, a genetic fusion, or amplification, we were looking for a very, very specific exon, which was at the 14th position and which was skipping alterations in the oncogene MET c-Met. So again, these typical challenges on the right-hand side you see are there, but what we were trying to do was trying to find these patients where we could only get the exon 14 skipping alterations. So it was kind of a hybrid study for us where we were dealing with advanced lung cancer patients, so we know time was not on our side or their health condition. But at the same time, we were doing something very unique, which was not done until at that time when half of the oncology or 3/4 of the oncology was focused on immuno-oncology combination studies. We were getting into this niche of a population with precision medicine. And the reason why I say this is that this is patient selection. And again, as I said, these were all the typical challenges, but I'm only going to focus on patient selection because it's really, really applicable in today's time as well. The lessons learned, the solutions we went after to get this patient. So here you will see why I keep calling this patient selection was the key for us, is that the c-Met exon 14 skipping population is only 3% out of the entire lung cancer population that we see today. And many of you might know that the lung cancer is the #1 cause of cancer death in the US as well as globally. And here we were going after needle in a haystack approach. And these were our challenges. So again, as I said, and this 3 to 4% of all cases globally, if you read this, the inclusion criteria correctly, it becomes clinically rate of more than 97%. Now, operationally, when we translate these to the investigators or the site, the first thing they are going to say is, oh, so we have to screen 100 patients to get just 3 patients for your study. We have to do so much work. And we got a lot of those questions, and you will see what we came, how we address those questions. So, a couple of key challenges were, yes, we only have 3 to 4% of these patients. But once we found these 3 to 4% patients, The other criteria for that was that they should have been chemo-exhausted, especially second or third line. So we were looking at very, very sick patients who were chemo-exhausted, had advanced lung carcinoma. And also, I would like to highlight that there was a very peculiar criteria that these patients were non-smokers. So you can understand the mindset of the families and the patients as well, that they were non-smokers and still were battling this disease, and again, chemo-exhausted. Another criteria, which was a translational medicine criteria for us, was to exclude EGFR mutation for us rearrangements, which actually is made up of, again, the remaining 97% of the cancer population. So again, we were going into the subset of patients, and time was not on our side because we had Tepotinib, which was a Pfizer drug ahead of us. But at the same time, NCI had an NCI match study was going on where major cancer centers in the United States were participating, and they were required to provide certain number of patients to that NCI match study, which was again NGS testing study ahead of of that time, I would say in 2015, 2016. So another challenge we had was that we had to, since this was extremely new, we had to find a central laboratory. So no local labs were allowed because we needed unbiased central confirmation of the net exon protein skipping status. Of course, since this was one of it, and we were just beginning, we had to find a companion diagnostics partner who would prepare this assay for us, who would prepare this NGS testing for us. And down the line, we also started discussing whether we should go for liquid biopsies, because again, we are talking about second, third line of treatment of patients, very sick patients, and their treatment might have started 3 or 4 years ago. So the biopsies that they did 4 years ago were not valid to come into our study. So the only option we had for them was to either give us a fresh biopsy, which was very risky proposition to the patient as well as to the investigators and sites, or versus go with a liquid biopsy where we could just get blood from their tumor and get the circulating tumor DNA and then develop the CDX partner and then just get the assay and get moving, confirm them. So all of this, we have to visualize how do we do this now. We needed 60 patients, and I think it was in Jay's presentation an hour ago, somebody asked that how do you decide a subset? I think it was Adriana who asked, how do you define a subset, or FDA gives you that number, that how do you define your N for the patient population? To answer that question, again, if she's listening, mainly what we did was, since even though we knew what we were up against, the efficacy data or the from the preclinical studies as well as from the phase 1 study was extremely promising, with 90% or more than 90% response rate and disease-free progression. So that was our cost to go to FDA and negotiate total number of patients. But at the same time, all we wanted was 10 patients in and get the futility analysis done so that we get that confidence level to go and ramp up this study globally. Because as you will see again in the upcoming slides, that getting this patients only in the United States was an uphill battle for us. So this is how we were envisioning patients coming into the Project Blue, we call. So it was, and also, pre-identification of patients and then the pre-screening. This was again a new thing for many investigators, and the funnel from pre-identification to pre-screening dropping patients in thousands. So from 2,700 pre-identified patients, we were getting into around 70 patients, and then from there, including other inclusion-exclusion criteria, we could get hardly 60 patients. And again, we decided that the cycle would be 21 days, and the only thing we required was just 4 weeks of cycle. That's 4 cycles, and that's it. 3-week cycle, total 4 cycles, and we will keep the operational burden once the patient is enrolled very, very low. Which I feel like in today's time, this criteria still exists for the sites, for the patients, to keep the operational burden low, to do all the work up here, pre-identification and pre-screening. And some of the methodologies that we used here at that time were all digital, which are again applicable today. And that's the reason I'm going back and forth between past and now. And I was explaining earlier that why it is important or critical. I saw that to mention is that these were the challenges, but again, this is the same thing that we are saying. Again, another depiction. So the 3 to 4% of screen failure rate was found actually in literature, because this was like the disease or the patient population which was completely exhausted. They had no hope whatsoever. And this is how the scenario was looking for us. That, okay, if we go with 98% screen failure rate versus 97%, if the worst case scenario for us was, what if we hit 99% screen failure rate? What if we only one patient qualifies after we screen 100? We still had to present this because, again, the background on the back burner was the timeline and the budget. That how are we, what is the burn rate going to be if we go after 99% screen rate? So it was very important for us to do all these exercises to balance all the access and the challenges of clinical trials that we were in, or this program, I would say. Strategies, and I would say we really had a brilliant team of scientists from Merck as well as from Quintiles, and I was actually working from both ends. So I was a program director at Quintiles, but then Merck approached me, and then I became Merck employee while I was working on this program. So I knew the teams from both ends, which was an advantage for me because then I could understand the scientific desire behind this, which, the brilliant scientists, but they were equally operationally nice. So my job was to constantly be in the middle to, streamline the communication. How do you convert this brilliance, scientific brilliance, into operational reality? Because when the rubber meets road, everything was going to a disaster. So, again, strategy consideration. So, I think Rachna in her previous presentation mentioned that, it has to be patient-centric here. One thing we learned was that there's no way we can open the traditional model where, okay, the sites are open, activated, and the patient walks in and, we know that the patient is CNET positive. No, or CNET alteration positive. So we had to keep, one thing we decided as a team was that we have to take the trial to the patient. Make it as easy for them as possible, and we do all the legwork again. So what we did was we sat on Google Maps, literally, and we identified community centers versus treatment centers and where we can go and get this and locate the central lab along with it, where if the satellite center of the central lab is near a major university hospital, that will be our treatment center, and we will take the study to the patients by opening just-in-time centers if needed. So again, this was our core strategy that, the targeted study sites to leverage patient populations and local testing for enrichment, and then parallel supporting strategies. And this is what I was saying, that this is the entire digital platform that we can utilize even today for rare diseases or such kind of patient populations. Where do you go? Everything digitalized. We only met these people once, but they kept referring patients to us through digital platforms. And this is how we envision finally the flow of patients, how the patient population we are going to tap into. Once they have the genomic testing through Foundation Medicine, we have reporting trial and matching services through N of 1 and Cure Forward. Then we have an entire network of physician and patient review. We also utilize what they call Enfusion, which was Moffitt Cancer Center's own database. Then consult central testing, then the qualified patient, and then we utilize TraumaTech to open the just-in-time site startup travel, and this whole thing we needed to tie up within 21 days of screening once we identified the patient. So I'm just briefly going to introduce you that many of you might know this, but this is something that we found at that time. Again, I'm talking about 2016, 2017, when, these were the emerging services which are now common. One of it was Cure Forward, where this is like literally a dating service, exact same platform copied for trials, clinical trials, where, a trial recruiter would just sit there and start matching the patients to the ongoing studies through clinicaltrials.gov. And it was offered free for patients to a certain number of trials, and it was also offered free to the sponsors and to the CROs because it was going through the pilot at that time, which we found just educating patients or just approaching patient advisory services to get there and they educate patients to enter their data was the key for this. Again, this is how it worked. Clinical trial exchange data with few patients, but then Cure Forward marketing picked up and they targeted around 25,000 members. And then there was this, no-cost pilot platform offered to everybody where, you just do the trial matching and the patient matching. The next one is also important, N of 1, and this is a local company here in Boston. But what they did was they had test annotation and reporting services for labs and networks. So they had a huge database, and they also had affiliation with US Oncology. So they were able to give us the footprints that of the geographical footprints of the patients. That okay, we have the annotated report. Here is what we see. There is your CMS positive patient. Go get them. And this is how they work. That they also had the trial match expansion for us and how they would work. And then we identified the players and the flow of communication. I'm just jumping here because I know I'm tight on time. But this is again, and from each of these services, my job was to, project the budget, the timeline, and also the enrollment rate. How much are we spending and how much can we get? So again, you can see, even with these efforts, we were getting like 1.5, which is ridiculous to put it this way, but yeah, up to 1 to 2 patients per month through these services. Same thing, similar to Cure Forward, this was trial reach that we went there and, very similar model, but again, they had good things. Foundation Medicine now is pretty famous. Everybody uses them. And Foundation Medicine at that time was applying for their Foundation One platform for FDA approval. So it was not approved by FDA, so we couldn't use it. So we had to go for a CDX platform with them. But they also promised, and they gave us pretty good numbers, actually. They told us that they see around 3,000 NGS tested cases every month. Even if they apply the exome coding skipping criteria, they would be able to give us 80 patients per month. Again, they applied the inclusion-exclusion criteria, and they were up to 2 patients per month, which we thought was pretty solid for us if we could get it. Intergen, again, same thing as I said, it was market database where we could just go and dip into and try to just understand where the patients are. Same thing, again, the impact, and all we were getting was 0.6 to 3 patients from Moffitt. We also went locally to MGH and understanding what they did, and MGH had a very interesting approach towards this. They said that, we are going to test only those patients who are negative for all other types of lung cancers, as I said, EGFR and ALK, will not go there, so we know what not to test. But then we had a really good super set of patients where we know, we just have to go focus on them and get them. This is just a map of all the centers who had a similar approach of NGS testing for patients before enrolling, and where those patients would look like. Where are they? So that it was easy for us to decide whether they would be treatment centers or just screening centers for us, and how many just-in-time sites we needed. And impact, so I'm going to jump on impact. So after contacting all these vendors, negotiating, creating the timeline, and everything for us was yesterday required within the 21-day screening timeline. So we decided, let's prepare a base case scenario, conservative scenario, and a worst case scenario. Even after getting all these things together and live for us, where do we go from here? And also, what if, even if the patient enrolls, but then again, there's a protocol compliance, where do we go from here? And what we realized was that once we started educating patients, once we started empowering them with all these digital services and assured them that there's everything we are going to do, the burden is on us, and also assuring the sites and especially the research nurses and everybody there that, we will do the heavy lifting of pre-screening. But once you get the patient, just don't lose them and make sure that you comply for the post-cycle requirement for us. We prepared this for our senior management presentation, that, okay, what if even after all these things, we don't deliver? So there was always this stress throughout this program. But once we started enrolling patients, like 1 or 2, within 2 cycles, we saw a definite positive response. The tumor was shrinking by almost 60%, which was extremely positive news for all of us, and which just kept going and going from there. So fast forward now, where are we? So Tepotinib and Skull got approved by FDA in 2021. We also got fast track approval because of the 10 patients futility analysis. We were able to negotiate with the FDA. And the overall enrollment was completed within 14 months. We expanded globally. And now this just-in-time and pre-screening patients heavily before you actually enroll them and sign a consent from them is something is adopted in Merck, and it was also an operational model for Avelumab, which is also now approved, and which is an immuno-oncology drug. So it started as a precision medicine approach in clinical operations, but it is also applicable everywhere. And lessons learned, again, as I said earlier and throughout, that first, communication, second, site and investigator network. It's just assuring everybody that the operational burden will be taken care of by the sponsor, by the CRO, because this is where I feel right now the communication is required. Because patients, especially in the COVID scenario now, as Rachna was saying, that patients are scared to go to the hospital. The hospital staff is exhausted. So what can we do? How can we step up for them? And there are numerous ways we can do this. We just need to always think that, okay, it's just not patients, but also the supporting staff that makes this clinical trial successful. They are also exhausted. So what can we do to help them as a team? And these are, the lessons that we shared across the programs, across the company right now, even within QuickEye. Every time there's a rare disease or new challenging cancer patient population is there, we always share details that testing is the key. Educating is another key for the patients as well as for the staff. But again, ensuring them that we are on the same team. It's not like our study, you go run with it, but no, we are equally responsible. We are going to do the heavy lifting. We will do the minimal operation compliance, and we go long time with that. So I'll stop now. Any questions? And again, thank you, everyone.
Great. Thank you very much for your terrific presentation, Monica. I'm actually a very big fan of the just-in-time activation of sites. I think that, IQVIA, Tempus, XCures is also developing models to be able to do this. And I think that, for these rare patient populations, it's really important. We do have one question in the chat. It's how difficult was it in the informed consent process to tell potential subjects that there was such a low likelihood that they would qualify? Did many potential patients decline because of the low probability of enrollment?
It's a very good question. So what we did was we had 2 kinds of informed consent. We had a screening informed consent where they knew that we didn't tell them about the study or anything. We just told them we are screening you for exon 14 mutations, that's it, and alterations. And then once they qualified that, then we gave them the option of the study because we didn't want their hopes to be really high that there is a drug that can cure them. But if they were not exon 14 skipping, then their hopes were crushed. So we were very careful in our communication that only screening centers will have the screening ICF. So we did not distribute it to the treatment centers at all, the screening ICF. So we were extremely careful how we communicate this to the patient.
Yep, no, I think that's a terrific strategy. Were the screening panels you did, were they broad or were they very specific for the exon skipping?
They were not broad. That was the problem because there was fusion, exon 14 fusion, alteration, overexpression. And so we kind of negotiated with our translation medicine team that if it's an overexpression and alteration, we will take them in.
Okay.
And the problem was that we had to be so specific is that we wanted to hit the futility analysis so we could get them fast track approval. And so we needed that data. So it was also driven by the statistical calculations behind it that we just couldn't go and offer it to all CMET14 or
Right. Now, I mean, my question is more around whether if you were able to offer a broader panel, would more patients have signed up for screening because they could potentially have had other options to if they didn't have the CMET exon skipping?
Brilliant question again. Yes, they did. And this is what we offered for the free of cost for the investigators, that we will get your patient everything screened, through NGS. So if the patient is ALK positive or TGFR positive, it's good to know. So it was an approach to just increase the NGS testing awareness in patients.
Yep. No, I think that's a terrific strategy. Again, very patient-centric and allows them to potentially benefit even if they can't enroll in your clinical trial.
Yes.
Yep. Okay. Well, again, thank you very much for your time and your presentation. This was really terrific. And so, Sam, I think we'll move on to the next speaker room. Is that right?
That's correct. Thank you again, Monica, for your presentation. I will be transferring everyone to the next room.